Okay. Good morning, everybody, and welcome to the Ceylon College of Physicians Specialty Update Series. This is the first of a newly redesigned Specialty Update Series that the college has introduced from this year. I'm Senaka Rajapaksa, the president of the college, and Dr. Tilak Jayalath, who is the president of the Sri Lankan College of Internal Medicine, will be joining us virtually from Kandy. From this year, the way this specialty update runs is that we will, for each month of the year, we collaborate with a sister college in the country. And this month, the collaboration is with the Sri Lankan College of Internal Medicine. The topic is a very timely one which we are all interested in doing. The vaccine has just been introduced, and some of our colleagues have already, who are sitting in the audience, have already had the vaccine and are still standing. The session runs for about 2 hours. We have an update on the vaccine for the first session with 3 eminent speakers joining us, followed by 2 lectures again on COVID-19. I will now hand over to Professor Tilak Jayalath, who is the president of the Sri Lankan College of Internal Medicine, to speak a few words and introduce the session.
Good morning. Welcome to this webinar on COVID-19 update. I think when you consider the date today in Sri Lanka, we are launching the vaccination program, and also we are having this webinar on COVID-19. So I think it's a very timely one, this webinar. And on behalf of our college, Sri Lanka College of Internal Medicine, I warmly welcome. We have 5 speakers today. Professor Malik Piris, Dr. Maheshi Ramasamy, and Professor Nilika Malavige, then Dr. Ananda Vijayavikrama, and Dr. Harsha Satish Chandra. And also we have some participants joining physically and virtually. So thank you very much.
Tilak, thank you very much, Tilak. I would like to ask Dr. Upul Disanayaka, moderator, to take the stage, and Professor Nilika Malavige is just about to start the first speech, but please wait a few moments for the introductions.
Thank you, Professor Sen Rajapaksa, President, Ceylon College of Physicians, and Professor Tilak Jayalath, President, Sri Lanka College of Internal Medicine. Ladies and gentlemen, welcome to the CCP Specialty Update on COVID in collaboration with SLSIM. Let me explain briefly the day's program. In the first hour, we have 3 world-renowned academics lined up who would speak on the vaccine for COVID-19, the truths and the myths. The speakers will be delivering their presentation one after the other, and the question and answer session will be afterwards. So kindly send the questions which will be posed to the speakers. The current therapeutic options and the case-based discussions will be after the Q&A session. And let me introduce the first speaker, Professor Nilika Malavige, who's a professor at the Department of Immunology and Molecular Medicine and the director of the Center for Dengue Research, University of Sri Lanka, and an academic visitor at the MRC Human Immunology Unit, University of Oxford. She is a member of the Executive Committee of the International Society of, and her main focus is infection and immunity. She has been studying the immunopathogenesis of dengue infection, vascular leak, and since recently, the correlates of protection and pathogenesis of COVID-19. She has won many awards for her research work and has secured many local and international grants. Nilika.
Thank you. I think it's thank you for inviting me to speak at this very historical symposium. I think the day that the COVID vaccine is introduced to Sri Lanka, and quite a few people sitting in the audience have received the vaccine. So I think it's a historical moment to speak about the COVID vaccine, and just to show what has been going on. So although we are introducing the COVID vaccine today, the rest of many other countries started introducing the COVID vaccine way earlier, actually in December. And as of now, Israel, I can't get the point out for some reason, but Israel has immunized 46 individuals per 100 people. That is 46% of the Israel population has been immunized. 27% of the Arab UAE population has been immunized, and around 10% of the UK population and so on. So millions of people have received COVID vaccines. In the US alone, 23.5% have received COVID vaccines. In UK, 7.6, and in India, around 2 million, because India rolled out the vaccine more recently. And what are these effects? I mean, what has been the success so far? We know that as far as vaccines were concerned, for polio, diphtheria, pertussis, we can't remember how it was like earlier, because most of us were not alive or very, very small children. So we have no idea the damage that the illnesses caused during those days, because when we were born, we were given the vaccines and we never saw that. So today we are seeing the effects of COVID-19, and let's see what the COVID vaccines do. So this is, again, the graph on your right shows in United States and US how the number of cases, not the deaths. Still they are experiencing quite a bit of deaths, because deaths occur way later. The number of cases have dropped after vaccination introduction into those countries. Of course, there could be many other factors apart from vaccine introduction, but this dipping of the number of cases actually was coincided with, that was after the vaccine introduction. And if you look at the graph on your left, so the light blue bars are individuals over 60, the dark blue bars are individuals between the 40 to 55 age group, and these are the proportion who were receiving critical care. So as you can see, after immunizing the over 60s, they were the first group to receive the vaccine in Israel, the proportion of over 60s in receiving critical care drastically fell. So this is the Pfizer vaccine, by the way, that was given in Israel, and you can see how it had already the signs of benefit in those countries which the vaccines have been rolled out. Now, a bit about the different types of vaccines. I won't talk about all the vaccines because Dr. Ramasamy is speaking after me, so she will speak in length about the Oxford vaccine, which most are receiving today. So there are inactivated vaccines, which all of us are familiar with, which we have received from childhood, where the whole virus is inactivated chemically or by other manners. And then we have, as far as this outbreak is concerned, the leading vaccines are the viral vector vaccines. There are several viral vector vaccines which I'll describe. And of course, the mRNA vaccine. So this is the first time the mRNA vaccines were introduced to the world and are doing quite well. And I'll speak about those. And then you have the recombinant protein vaccines. These don't have any genetic material, but contains the main viral proteins for which individuals develop an immune response to. Now, how do mRNA vaccines work? The reason I think it's important to highlight why mRNA vaccines work is because of the myths going out. So mRNA is a messenger RNA. So it is incorporated in a liposome, and when you inject it, the lipid nanoparticle takes this mRNA inside to the cytosol. So there we have ribosomes, and ribosomes translate messenger RNA. You would have learned that in A-level. So the messenger RNA translates the RNA and converts into a protein. So that happens in the Golgi apparatus. And so then you get the spike protein, because these mRNA vaccines only contain the spike protein of the virus. And this is RNA. And our genome is DNA. There is no way that the RNA incorporates into our genome. That is impossible. So our cells make ribosomes of our cell translate this the RNA into protein, making the spike protein, and then when it is excreted out, you develop antibody responses to this spike protein. And also in the cytosol, they are put on these MSC class 1 and class 2 proteins and presented to T cells. So you get a robust CD4 and CD8 T cell response. And the two mRNA vaccines that are around are the Pfizer and the Moderna. The Moderna vaccine, I think, is not going to be available elsewhere apart from the US. So I will speak briefly about the Pfizer vaccine. So the phase 3 clinical trials data was published in the New England Journal of Medicine of Pfizer. And as you can see, there is a huge difference in the incidence of infection. So when you say incidence of infection, in these first generation vaccines, COVID-19 vaccines, what was important, or the WHO endpoints, was reduction in severe and symptomatic infection. So the endpoints of these vaccines were reduction in symptomatic illness, and as you can see, there is a drastic reduction in those who develop symptomatic illness in those who receive the Pfizer vaccine. And this differentiation is seen after 12 days and not before 12 days. The reason it takes 12 days after the first dose is you don't get an immune response the next day after you are vaccinated. So it takes time for the immune response to develop. But you see this separation in the efficacy after 12 days. So the efficacy after the first dose is around 52%, and after the second dose, it goes up to 95%. And the main side effects were pain at the site of injection, headache, and fatigue. So I think adults feel these more than children, because most of us have received the vaccines when we were kids, and we couldn't complain about any of this to our parents. We couldn't talk. But basically, so pain at the site of injection and systemic signs such as headache and fatigue. And these were more prominent in the young age groups than the old age groups. But of course, there are concerns about side effects for this vaccine, because anaphylaxis and allergic reactions were noted in some individuals. So this analysis, which was done over a period of 10 days, from December 14 to 23rd, 2 million individuals received the Pfizer vaccine, and 21 individuals developed anaphylaxis and 83 developed allergic reaction not amounting to anaphylaxis. So the risk of anaphylaxis was 11.1 cases per million doses, which is 10 times more than a usual vaccine. And that is thought to be not confirmed due to this pegylated component. And 81% who developed anaphylaxis or allergies had a documented history of allergy or anaphylaxis, and the non-anaphylactic reactions were characterized by pruritus, rash, and mild respiratory symptoms. What I would like to draw your attention is to the second column, where the number of individuals, the gender of the individuals of who developed anaphylaxis and non-allergic anaphylactic reactions is 90% are women. So yes, I know Dr. Pool is looking at me in a strange way. Yes, so I mean, ideally, both men and women are supposed to develop anaphylaxis equally, but 90%, the anaphylactic reactions were 90% in women, and the non-anaphylactic allergic reactions, again 90% were in women. So I don't know, I don't think anybody knows the answer why these allergic reactions are more frequent in women compared to men. We will hopefully find out the answer one of these days. And so coming to the inactivated vaccines, there are three leading inactivated vaccines: the Sinovac, all Chinese, Sinovac, the Sinopharm, and another one without, which I couldn't actually get the name, developed by the Wuhan Institute of Biological Products. And so the phase 1 and early phase 2 data has been published for the Sinovac and the Sinopharm vaccines, but any data, early phase 3 data or interim analysis of these vaccines have not been published, so we can't comment about the safety and efficacy in the context of a phase 3 trial. And coming to the viral vector vaccine, so here the main vector here is the adenovirus. It is either chimpanzee adenovirus or a human adenovirus, and most of the platforms use a non-replicating virus. Now the reason for this is, as far as COVID is concerned, it is those with comorbidities and certain types of immunosuppression that have severe disease. So you want to vaccinate those age group, those individuals. So this vaccine can be given to those immunocompromised individuals because it's a non-replicating virus. So in this virus, you have the spike protein inserted. So one of these vaccines is the Oxford AstraZeneca vaccine, which is produced in 8 manufacturing plants all over the world. And Serum Institute is one of these manufacturing plants, and it's the vaccine that was given to you today. The other one is Sputnik 5, which is the Russian vaccine. The phase 1 data has been published, and early phase 2 data, but there are no publications beyond that. So we don't know the efficacy and the safety of the Sputnik 5 vaccine again. And the other ones, so this morning, the other one, the phase 3 data was published, interim analysis, which looked good in the Novax. Now, Sri Lanka has a very proud history of successful vaccination. Our vaccination campaign is possibly the most successful campaign in the whole world, which with over 98% or even more, 99% vaccine intake. So, and people have very much faith in vaccines. So it is very important that we don't make anything go wrong with our vaccination campaign, and it is absolutely essential to take safety measures, because after all, you are immunizing healthy individuals. So the quality is important. So this, if you have time, it will be good to look at this paper in Nature Medicine. And of course, quality is a really important issue. There should be no difference between batch-to-batch variations, the standard procedures. And with upcoming all these vaccines coming up, there should be proper regulatory pathways to give authorization for vaccine use. Now, the WHO has named the USA, UK, Europe, and Japanese regulatory authorities as very stringent regulatory authorities. So when vaccines are evaluated by those authorities, they take into a lot of things account. Now, Sri Lanka has not been in so far, the vaccines we have got and are given to us since childhood, they have been approved by the WHO, approved by all those bodies before we got it. So we have to be very careful to decide which vaccines we will give authorization to or not, based on this very important issue of safety. Now, for instance, this is from this journal article that I showed. In the United Arab Emirates, they have approved a COVID vaccine without any safety data or efficacy data. So I think, I don't know if Sri Lanka would take a decision like that to approve vaccines without efficacy data. And then the next question is, how many individuals should be immunized to prevent transmission? So the so-called herd immunity, because I know some countries went for the approach of natural herd immunity, which had devastating consequences. But if there's a vaccine to achieve herd immunity, that would be the answer as we are seeing now. And for measles, pertussis, polio, they are very high basic reproduction numbers, so you need a very large herd immunity. For the SARS-CoV-2, it has been predicted about 60 to 83% of the population need to be immunized for herd immunity, and the vaccine efficacy should be between 60 to 95% to achieve that target. And finally, who gets it? So right now we have only limited doses. We got 500,000 doses, enough for 250,000 people as a gift from India. So the first in line are healthcare workers at high risk, and if we have more, the end-stage kidney, stage 3 and 4, any age, and then elderly with comorbidities. And if we have more vaccines, then it would be the frontline workers, all frontline workers, and of course kidney disease, and people over 60 with comorbidities. If we have more than 20%, then it will be everybody more than 60 years, individuals with comorbidities less than 60 years, and if we have enough to go around, then of course population at high risk working in factories, economic centers, and prisons, because it's important to keep the economy going for various reasons, and schools and educational institutes functioning. Okay, thank you. I'll stop there. I think Maheshi will continue from there. Thank you.
The next speaker is Dr. Maheshi Ramasamy. Dr. Maheshi Ramasamy obtained her medical degree at Rice College, University of Cambridge, and trained in infectious diseases and general internal medicine. She obtained a DPhil at Wadham College, University of Oxford, and is currently a consultant physician at John Radcliffe Hospital in Oxford. Her research interest is in adaptive immune responses to vaccines, and she is a principal investigator at the Oxford Vaccine Group, where she leads on COVID-19 vaccine clinical trial. Dr. Maheshi Ramasamy.
Good morning, and thank you very much for inviting me here today. I'm delighted to be speaking to you today when the very first Oxford AstraZeneca vaccine has been administered to the first person in Sri Lanka. I'm just going to share my screen. So I'm going to give you a bit of an overview of how we managed to develop the Oxford AstraZeneca vaccine over the last year. As you know, it normally takes up to a decade to develop a vaccine, right from the initial concept up until licensure. And that's because there are a variety of steps that need to happen along that way. We need to generate preclinical data. We need to undergo phase 1 studies to look at safety. We need to look at phase 2 studies to look at dose escalation or age de-escalation. We then need to do phase 3 studies to look at efficacy. We also need to manufacture the vaccine in a way in which it's safe to administer to humans. We were fortunate to be able to expedite the development of the vaccine over the last year for several reasons. Firstly, because we had a huge amount of funding from the UK government that allowed us to be able to use several of these phases in parallel. We had a huge amount of support from researchers in the UK and around the world. We had manpower to deliver the trial. And we also worked very closely with the regulators right throughout. So we were providing data to them as we went along so they could review the data in a timely way. Now, our vaccine, as Professor Malavige has already said, is based on an adenoviral vector. So this is a virus that doesn't normally cause disease in humans. It normally infects monkeys. And what we've done is we've taken the gene encoding the spike protein in COVID, and we have engineered it into the DNA of the adenoviral vector. And this virus absolutely cannot replicate in humans, and that's because it's been genetically modified. We've taken out the essential genes needed for viral replication. In fact, the only way that we're able to grow this virus in culture during the manufacturing process is to grow it in a transgenic cell line that contains these extra genes. This vaccine is stable under normal cold chain circumstances. Because it is non-replicating, it is absolutely safe to use in individuals who are immunocompromised. So how does it work? Well, as we've alluded to, we take our gene of interest, in this case, the S gene, we clone it into our adenoviral vector, and then we use the vector as our vaccine. Once that's been injected into muscle cells, the DNA from the virus is transcribed into messenger RNA, and then mRNA for spike protein is translated into the protein itself. The viral genetic material degrades after a couple of days. However, the protein can then be trafficked to the cell surface and expressed on MHC, presented to T cells, or taken up by antigen-presenting cells to stimulate humoral or antibody-mediated immunity in the lymph nodes. Once we developed our vaccine in February, we were then able to move very quickly to animal studies. So this is data showing what happens to rhesus macaques when they are immunized with our vaccine. And we're able to generate really good neutralizing antibody titers in monkeys. We were also able to generate good T cell responses in monkeys. After we'd vaccinated the animals, we then challenged them with live SARS-CoV virus. And as expected, the control animals, and this is a lung specimen from a control animal, had florid pulmonary infection, whereas the vaccinated animals actually developed very little disease and were protected. So that was very encouraging. And as soon as we had that safety and immunogenicity data from the monkeys, we then moved into humans, and we started our phase 1 trial in Oxford. This recruited healthy adults. We had over 1,000 participants, and we gave them a single standard dose of the Oxford vaccine. This was a randomized, controlled, blinded study. So our participants didn't know which vaccine they had. They were randomly allocated to have either the Oxford vaccine or a quadrivalent meningococcal vaccine, the MenACWY vaccine. And again, it was encouraging that in humans, we were able to generate very robust neutralizing antibody responses. So people were vaccinated at day 0, and we saw these good neutralizing antibody responses after a single dose of vaccine. And the responses that we saw were not dissimilar to the kinds of neutralizing antibody titers that we saw in people who'd recovered from COVID-19, so in convalescent samples. People who were immunized with MenACWY, as expected, didn't develop any antibodies at all to spike against COVID-19. What was interesting is that we had a few participants who were seropositive at baseline, which means they probably had already had COVID by the time they were vaccinated. And actually the vaccine boosted their immune responses, as you can see here. We also were able to show that we generated good T cell responses with the vaccine. And here you can see T cell responses peaking at 14 days after vaccination. We then moved on to our phase 2 study, and here we were testing the effect of the vaccine in people of different ages, 18 to 55, 56 to 69, or over the ages of 70. We used 2 doses of vaccine here, and people were immunized 28 days apart. So they had a prime on day 0 and then a booster at day 28. And we recruited several hundred people from each age group. We used 2 different dose regimes, so either 2 low doses or 2 standard doses. I don't have time to go into the reason behind the choosing of this, but I'm happy to take questions on it at the end. So one of the things we were very interested in looking at was how well tolerated was the vaccine, what sorts of symptoms did people have immediately after vaccination. And so all of our participants kept an electronic diary for the first 7 days after each vaccine, where they documented what sorts of symptoms they had. And we were looking at local symptoms, so things like pain in the arm, swelling in the arm, or systemic symptoms, so things like fevers, muscle aches and headaches and so on. So in this slide, each column represents a different systemic symptom. So for example, here we have fatigue, fever, joint pain, and then the different rows represent the different ages. So at the top, we have the 18 to 55-year-olds, in the middle, 56 to 69, and at the bottom, the over 70s. And there are a few things to take home from here. Firstly, that the sorts of symptoms that we saw were the kinds of things that you expect after any vaccination. So, sore arm, feeling a bit tired and a bit fluey, but actually all these symptoms lasted only for a couple of days after vaccination and then resolved. The majority of the symptoms were mild. And what was interesting is that actually older people had far fewer symptoms than younger people. And this was true of individuals who received the high dose or the low dose. And when we looked at the second dose of vaccine, actually everybody, regardless of age, had fewer symptoms after the second vaccine. We looked at neutralizing antibody titers in these individuals as well, and that's what you can see here in the panel on the left. So the different colors of this graph represent people of different ages. On the top are people who had 2 standard doses, on the bottom are people who had 2 low doses. In the dotted lines are people who had a single dose of vaccine, and people who had 2 doses were boosted here at day 28. And the take-home message from this is actually that regardless of vaccine dose or age, most people had very similar neutralizing antibody titers by 28 days after booster. And that was very encouraging. We also, and clearly 2 doses elicited higher neutralizing antibody titers than a single dose. We also saw very robust T cell responses at all ages, and as expected, T cell responses peaked at day 14 after vaccine. One of the things we were concerned about was the concept of anti-vector immunity. Might the immune response to the adenoviral vector itself impact on anti-spike responses? So in this top graph here, I've plotted anti-vector antibodies over time, and you can see that in people who received 2 doses, there was no boosting of the anti-vector antibodies after the second dose. So that was quite interesting. When we then plotted the anti-vector antibodies against either neutralizing antibodies to COVID, to SARS-CoV-2, or to T cells, we saw that there was no correlation between anti-vector antibodies and immune responses to the spike protein. So that was reassuring and suggested that it was safe and effective to continue to use 2 doses of the adenoviral vector. By June, we had moved on to our phase 3 study. So here we were, as well as looking at safety and immunogenicity, starting to look at efficacy. So this means clinical protection from COVID disease. Now, in order to see a signal, a statistically significant signal for efficacy, we needed to have a bigger sample size. So we expanded our trials to include up to 23,000, over 23,000 participants. in the UK at this stage, we were starting to, as a result of lockdown, the number of cases of COVID from our first surge was starting to fall. Now, in order to show efficacy from disease, you have to have enough disease circulating for vaccine to prevent it. And for this reason, we opened trial sites in Brazil and South Africa who were just starting to see the onset of their pandemic there. So in Brazil and South Africa, we used a standard dose regime. And in the UK, we had a low dose, standard dose regime. We presented our interim analysis results in November last year, and by this time we had accumulated over 70,000 person months of safety, with each participant being followed for a median of about 3.5 months. Again, we saw very similar local and systemic reactions to what is seen with, say, for example, a flu vaccine. One of the other things we looked at was something called serious adverse events, or SAEs. Now, these are events that are either the diagnosis of a significant medical condition or a condition that requires hospitalization or can lead to disability. Now, when you've got a trial of 23,000 participants, serious adverse events happen. People get heart attacks, people get appendicitis, people are in road traffic accidents. And in each of those cases, we had to have an independent safety review to see, could that serious adverse event possibly in any way be related to the vaccine? We saw that there were very similar numbers of serious adverse events between the control and the Oxford vaccine group, and details of all of these are available in our Lancet paper. The majority of them were judged to be not related to the vaccine. However, we did have 3 possibly vaccine-related SAEs. So one was a case of hemolytic anemia, that was seen in a participant who'd received the control meningococcal vaccine. One was a case of transverse myelitis. So this was in somebody who'd received a second dose of the Oxford vaccine. This was a short segment of demyelination, and the patient recovered after several weeks. And then we also saw a very high fever, a single case of a very high fever in a patient who remained blinded as to vaccine allocation, and they received a second dose of the vaccine without having any similar responses. So the independent data and safety committee, as well as the drug regulators, reviewed all of this safety data, and they felt that the vaccine had a good profile and subsequently gave approval on this basis. Our co-primary endpoint was efficacy. Now, when you're defining efficacy, we looked at it as prevention of symptomatic disease by the vaccine. So we defined our endpoint as people who were PCR positive and who had either a cough, a fever, breathlessness, or loss of taste or smell. We did also look at other exploratory endpoints, but I don't have time to go into those here at the moment, but I'm happy to take questions on them afterwards. So this is a graph of the incidence of COVID infection, symptomatic PCR positive COVID infection over time after the second dose of vaccine. And in blue are individuals who received the the control vaccine, and in red are people who received the Oxford AstraZeneca vaccine, with the dotted lines representing 95% confidence intervals. And overall in our trials, we saw 70% efficacy against symptomatic COVID disease. Importantly, in consistency with the WHO criteria for an effective vaccine, we saw no deaths related to COVID-19 in people who'd received our vaccine. We had no severe cases of COVID-19 infection as judged by the WHO severity criteria, and we didn't have a single hospitalization of any of our participants with COVID-19. So that was really encouraging. One of the things that generated a lot of interest was the difference between the efficacy that we saw in people who received low dose, standard dose versus standard dose regimes. And I just thought I'd spend a few minutes just quickly unpicking that here. Actually, when we went back and looked at the data, we felt that the difference in efficacy, we apparently saw a higher efficacy with the low dose standard dose group. That difference was actually more likely to be related to dose interval between the first and the second dose. So in the UK, because of issues with vaccine manufacturing, most of our participants had in the phase 3 study actually had quite a long interval between their first and the second dose. So of the people who'd received low dose standard dose, most people had a dose interval of between 8 to 16 weeks. And similarly, even in the standard dose group in the UK, we had a dose interval around over half of our participants had a dose interval over 8 weeks. However, in the Brazilian trials, the majority of participants had a 4-week dose interval. So what does that actually mean? Well, when you look at the absolute antibody titers of people and compare it with dose interval, what we saw was that with an increased dose interval, you have better antibody titers. So in this table here, this is the antibody titer of people who had a 12-week interval between their first and their second dose, and it's about 3 times higher than the absolute antibody titer for people who had around a 4-week interval. So this data was used by our drug regulator to recommend a dose interval of between 4 and 12 weeks on the licensing, on the emergency use authorization approvals. And the UK government has taken the pragmatic decision to offer people a vaccine at a 12-week interval, partly due to this data that we have actually improved antibody responses, and partly to allow equitable supply to allow as many people as possible to have that single first dose. This increase in immunogenicity with dose interval is something that we see with other vaccines too. So for example, with the hepatitis B vaccine, we know that a 6-month dose schedule is actually more likely to give you higher titers of antibody than a 1-month vaccine schedule. When we did a post hoc analysis looking at efficacy by dose interval, we did also see a trend to higher efficacy with an increased dose interval. So based on all this advice, the safety and the efficacy data, the MHRA, the UK drug regulator, approved the use of the Oxford AstraZeneca vaccine on the 30th of December last year, and many countries have since followed suit, including Sri Lanka last week. But what does this actually mean for us here in the UK? Well, just a few days ago, we passed a very grim milestone of over 100,000 deaths from COVID over the course of this pandemic. Although cases are starting to fall, we're still seeing, tens of thousands of cases a day, although the effects, as I said, of the lockdown are starting to be seen, and we are starting to see a bit of a fall. However, we still have huge numbers of people in hospital, and the National Health Service is incredibly stretched in dealing with these patients, and many routine procedures and operations and treatments are being postponed or delayed. However, the light at the end of the tunnel is that we've vaccinated almost 8 million people in the UK. About a third of these have received the AstraZeneca Oxford vaccine. We have a very robust post-authorization safety surveillance mechanism in the UK run by the Medicines and Health Regulatory Authority. And we have had no post-authorization safety concerns reported by the MHRA, which is very encouraging. I'd just like to finish off by saying that this has been an absolutely enormous team effort. A lot of people working very hard, both in the UK and overseas, to deliver these trials at pace. We're grateful to our funders who've allowed us to deliver these studies. We're grateful to our partner in manufacturing, AstraZeneca. But most of all, we are grateful to our trial volunteers, over 23,000 people who volunteered to take part in our trials, without whom we would not have this data, without whom we would not have a vaccine that is in use today. Thank you very much.
Is Professor Malik Piris. Professor Malik Piris is Professor of Virology at the School of Public Health at the University of Hong Kong. He is a clinical and public health virologist with a particular interest in emerging viral diseases, animal-human interface, including influenza, coronaviruses, SARS-CoV, MERS-CoV, SARS-CoV-2, and others. In 2003, he played a key role in the discovery that a novel coronavirus was a cause of SARS, its diagnosis and its control. Currently, he is researching the recently emerging MERS coronavirus and most recently, the newly emerged SARS-CoV-2. He has over 650 peer-reviewed research publications in international scientific journals. For his research contributions, he was elected a Fellow of the Royal Society of London in 2006 and Foreign Associate of National Academy of Sciences in 2017, which are two most prestigious scientific organizations globally. He will be speaking on coronavirus vaccines, the knowledge gaps. Over to you, Professor Malik Piris.
Thank you very much. Can you confirm that you can hear me and see my slides?
We can hear you. We are up yet to see your slides.
I have
Now,
shared
yes.
the screen. Yeah,
Yes,
okay.
we can see you now. Yeah.
Yeah, so let me join the other speakers in congratulating Sri Lanka in starting the vaccination campaign today. That's fantastic news, and thank you for inviting me to take part today at this meeting. So I will address some of the knowledge gaps that we still need to understand regarding vaccines. And this includes the duration of protection after vaccination, what are the measurements, immune measurements, antibody or any other measurements that correlate with protection? Do vaccines prevent transmission? You heard already that vaccines do protect against disease. Then, of course, something in the news recently, these virus mutants or variants. And finally, something that I understand concerns some people in Sri Lanka about this antibody-dependent enhancement of viral replication. So firstly, the duration of protection. Now, clearly, the vaccine clinical trials started only a few months ago, so we don't have full data on long-term duration of protection, or for that matter, of antibody responses. So that is something that needs to be defined. However, what we have been doing is to look at natural infection and look at neutralizing or protective antibody titers after natural infection. And what you see there is that in patients who have recovered from COVID, you do have antibody lasting up to 200 days and or longer. So essentially, when you stratify these patients by severity, patients who are severe and mild have very good antibody titers lasting for well over a year. And asymptomatic patients also have good antibody titers, but the duration seems to be shorter. So that may be an indication that vaccines also, which, as you heard, generate antibody responses comparable or sometimes better than natural infection, should at least provide that degree of protection, that duration of protection. Now, I should also point out that just because antibody titers wane over time and go to undetectable levels, it doesn't mean you're not protected. So for example, we know that with hepatitis B vaccination, as long as you make a good antibody response at the start, even after many years when your antibody titers are not detectable, you're still protected because when you meet the virus, you have a memory response, a booster response. And you have to also keep in mind that it's not only antibodies, T-cell immunity, for example, would be important. And we know that after SARS in 2003, people who recovered from SARS still have good T-cell responses, even though their antibody responses have waned. Then what are the immune correlates of protection? What can we measure to know whether somebody is protected? So now there's increasing evidence that previous infection with COVID protects from reinfection. And we know that serum or antibody can protect from infection or challenge, because in experimental animal models, when you just transfer serum from an immunized animal to another animal and you challenge that animal is protected. And in the case of monkeys or primates in vaccine studies, the protective antibody has been shown to be neutralizing antibody predominantly, although some other types of antibody also may be protective. And in the neutralizing titers, the development of neutralizing titers are correlated with reduction in viral load. But what we don't know so far is what titer, what neutralizing antibody titer would be the limit of protection. So for example, for influenza, we know fairly well that a hemagglutination inhibition titer of 1 in 40 is correlated with a 50% protection in the population, but we don't have that same information for COVID. I mean, clearly these are information that will come over the next few months. And similarly, we don't know the role of T cell responses and mucosal antibody in protection. Now, a big question is, do vaccines prevent transmission? As you heard from the previous speakers, the vaccine trials of necessity are measuring protection from symptomatic disease and not prevention from transmission, because that's an extremely difficult thing to measure. But there is some data that is becoming available in this area. So on the basis of the current clinical trial data, we cannot just assume that just because a vaccine has a 70% protection from symptomatic disease, it means that there's a 70% protection from transmission. It may be some level of protection, maybe no protection. I don't think that is the case from transmission, I mean. So if you look at other vaccines, for example, if you take a vaccine like measles, we know it has is very effective in protection from disease as well as from transmission, because it provides sterilizing immunity, meaning there is no asymptomatic infection as well as symptomatic infection. But other vaccines like whooping cough will protect you from disease, but not necessarily prevent you getting infected in the nasopharynx, and therefore you might transmit to others. Now, in experimental monkey studies, the vaccines, number of the vaccines showed that, of course, the lungs are protected. The nasopharynx, the virus load is reduced, but not completely eliminated in some of the vaccines. In some vaccines, it was actually eliminated. So the data that are awaited are, for example, do vaccines prevent asymptomatic infection? Now, this is very challenging. The AstraZeneca do have some data in that regard. And do vaccines have a reduction, even if people develop infection, is the viral load reduced, which would indicate a possible reduction of transmission? And of course, now you heard that Nitika talking about the very high vaccine coverage in places like Israel. So we will, in the next few weeks and months, be able to see whether that high vaccine coverage is resulting in also reduction of transmission to other people who are not yet vaccinated. So I think that data will come. But I think in the meantime, of course, we have to be cautious. So we do need to maintain other measures, even though we have been vaccinated, such as social distancing, wearing masks, etc. And also, I don't think it automatically means that just because you're vaccinated, you will be allowed to travel internationally without quarantine or whatever it is. So for the moment, we have to be cautious in that regard. Then, of course, the question of viral mutations. So viruses replicate inside cells, and RNA viruses do make errors in their replication. So the viruses continue to mutate. Now, many of these mutations are damaging to the virus, so the virus dies, or they don't make any difference. So but a few of them can actually provide an advantage to the virus. So those are the ones that we are concerned about. And particularly, we are concerned about this very early stage of the virus attaching to the receptor, because this is where the vaccine immunity also works. And this basically shows schematically enlarged picture of that same site, where this is the cell and this is the virus spike protein binding to the receptor. Here, this is the virus receptor shown here. And this surface of the molecule is the part that binds to the host cell. And so mutations in this area is particularly important in terms of escaping from neutralization and maybe also enhancing transmission. So very, very quickly, as you know, there is a particular variant called B117, which was first detected in the UK, which they have shown is 50 to 70% more transmissible than the previous virus. But fortunately, this particular virus is not escaping from immunity, either from natural infection or from vaccines. So the vaccine should work very well against it. Now you have to point out, I have to point out really that the fact that this mutation was detected in the UK doesn't mean it arose in the UK or that other countries don't have it. You will detect something only if you look for it. And UK and South Africa and a few countries have been doing a lot of virus sequencing, which is why they have detected some of these mutant viruses. So similarly, in South Africa, again, a separate emergence has been detected. And here, there is evidence of increased transmission, but no evidence of increased severity. But here, there's one particular mutation which one would have expected to have an impact on antibody neutralization, and that is indeed what they do see. Now, you can see the impact of increased transmission by this yellow bars here. These show the percentage of this mutant virus, which is this yellow virus, if you like, over time, over the months of October, November. You can see it was hardly present in August, September, and then very rapidly, it has taken over as the dominant population of virus infecting people in South Africa. Now, again, another mutant of concern has arisen from Brazil, and the concern is that it shares one of the same mutations as the South African virus. So what is the impact on neutralization with antibody? So this is the B117 or the UK detected virus, and you can see the wild type virus and the mutant virus essentially gives similar neutralization titers. So the mutation is not affecting protection from antibody. The South African virus, which has this particular mutation, you can see there's reduction in neutralizing antibody, but it's not completely gone. So it is compromising protection a bit, but not complete loss of protection. So just to summarize about the viral mutants, some of these mutants, particularly the 484K mutation, which has been found in South Africa and in Brazil, is impacting neutralization, but not completely abolishing it. And also keep in mind that neutralization is not the only mode of protection. There is other antibody and T-cell mediated immunity that can contribute. And already some of these vaccines, particularly the RNA vaccines, are already being updated or modified to cover these mutations. And also keep in mind, of course, these mutant viruses do not transmit in any different way. They are transmitting in the same way. The same social distancing measures should work. But of course, we have to work harder to achieve the same level of control. Now, the question of whether we would need yearly vaccination like we do with influenza, I don't think it will be as bad as influenza, and we can discuss this later in questions. Now, the question of antibody-dependent enhancement. So first and foremost, let me show you what we mean by antibody-dependent enhancement. This is, of course, particularly relevant for dengue. And indeed, my PhD thesis was on antibody-dependent enhancement going back 40 years ago. And what you see here is another mosquito-borne virus, West Nile virus, because that was easier to work with for these particular studies. So what you see, each of these holes you see, these white spots, are a single virus causing infection and killing the cells. So you can actually count the number of infectious viruses here. Now, if there is neutralizing antibody, this should be completely protected. So there should be no holes. Now, when you put in a cross-reacting antibody, which is not neutralizing, what would you see? You would expect to see no change. But in this particular case, and these are macrophage cells, you can see that with cross-reacting antibody, you have huge increase in viral replication. So this is what is meant by antibody-dependent enhancement of viral replication. Now, crucially, as I told you, these are macrophages. And of course, more recently, this has been shown to be possibly contributing to the enhanced disease in the dengue vaccine story. Now, how this works is under normal circumstances, the virus attaches to the cell, gets into the cell, replicates in the cell, and you have thousands of viruses leaving. Now, if you do have antibody in that is binding to the virus but not neutralizing it, if this cell is a macrophage, there is a second path of entry into the cell through these Fc receptors which bind virus antibody complexes. Now, so for antibody-dependent enhancement to take place or to be relevant, firstly, the cells need to have Fc receptors. So that means they are monocyte macrophages largely. These macrophages need to be permissive to virus replication and be the important target cell in natural infection with that particular disease. Now, in the case of dengue, all these are true. But in the case of COVID-19, we have shown in our own lab that these human macrophages can get infected with COVID-19 or SARS coronavirus 2, but they do not replicate infectious virus out. And secondly, of course, macrophages are not the primary target cell of replication in COVID-19, unlike in dengue. So I would confidently say that it's highly unlikely that antibody-dependent enhancement will be relevant in COVID-19. And it has not been observed in other preclinical or in clinical trials so far. So I've tried to cover all these questions, and I will be happy to thank you for your attention on behalf of my other co-panelists as well as myself, and we will move to questions and answers. Thank you.
Now the question and answer session. We are getting a lot of questions from the people who have connected virtually. The first question goes to Dr. Mahesh Ramasamy. The burning question is, most of our healthcare workers, at least a fair percentage of the healthcare workers are breastfeeding women. What do you know? It is contraindicated, but the question they're asking is, if they stop breastfeeding for a while and get the vaccine, would it be okay?
Thank you. That's a question that I get asked quite a lot here in the UK. At the moment, there is no data on the safety of the vaccine in breastfeeding. There's no reason to suppose why there would be a problem. The Oxford vaccine doesn't replicate, it can't give you COVID. We don't expect to see spike protein in breast milk. So there is no reason to suppose that it would be a safety problem. It's just that the data as yet is not there. So the UK regulators have taken the pragmatic approach that it has to be an individual decision. So if a person is at increased risk of COVID disease, so for example, because they have underlying comorbidities, or because they are at increased risk of acquiring disease because of their job, they're a healthcare worker, for example, then that person needs to take an individual decision as to the theoretical unknown risk against the actual risk of having COVID, of getting it themselves, of transmitting it to their families and to their patients. So it's very much got to be an individual decision, but there is no data.
Thank you. And again, the question goes to Dr. Ramasamy. Why do you suppose that pregnant women should not take the vaccine?
Again, this comes down to lack of data. So when we, with all these early human phase clinical trials, the drug regulators require us to in phase 1 and phase 2 trials to not recruit people who are pregnant or who are planning to get pregnant. And that's out of an abundance of caution while there is no data. So when we enroll people, we ask them to use contraception for the duration of the study. Of course, there are people who got pregnant during our study inadvertently, and we will be following them throughout the duration, so for over the course of their pregnancy, and then after their babies are born, to look for any signs of any safety signals. There are formal pregnancy studies planned by AstraZeneca. The advice for someone who's planning to get pregnant or who is pregnant, again, is to think about what your individual risk is. Does the theoretical risk of vaccination outweigh the actual risk of developing COVID? Again, the vaccine cannot replicate, it cannot cause disease in the mother or the baby, it cannot cross the placenta. And I have many pregnant colleagues who have actually been vaccinated because the risk to them of getting disease is far higher than this theoretical risk.
Uh, Again, the question is to Professor Ramasamy. Now, some of the studies say that the Oxford vaccine, the effectiveness is 60% and some say 94. So why do you see a range of effectiveness of the vaccine?
So that comes down to the difference between dose intervals. Our overall efficacy, when you pool all the data, is 70%. As I explained, in the UK, for logistical reasons, for vaccine manufacturing reasons, we ended up having quite a long gap between our first and second dose of our participants in the phase 3 component of the study. And actually, the longer the gap between the first and the second dose, the better the antibody response. And that's actually what the difference in efficacy was down to. It was due to the fact that people who were having a longer interval between the first and the second dose actually had better antibody responses and were therefore likely to be better protected.
Thank you. The next question goes to Professor Neelika Malavige. Now, if a person has had COVID infection in the past and recovers, what are the chances of that person getting COVID again without a vaccine? Is it possible?
Yes, I think it is possible. The studies have shown reinfection, but not a lot of reinfection. It is very, very limited. But especially people who are very much exposed to the virus regularly, like healthcare workers, the risk might be higher. And people who have had asymptomatic infection, as was shown, their antibodies may wane, but still we don't know the risk of infection because antibodies are not the only mode of protection. So as of now, we don't know the proportion of individuals who can get reinfected. But because we don't know that, the advice is, even if you have had COVID, to get the vaccine.
So the advice is, even if you have had COVID in the past, you should take the vaccine if your chances are higher. So how long after the so-called cure of COVID should you take the vaccine? Could Dr. Mahesh Ramasamy answer that question, please? Now, if you have had COVID in the past, how long after the recovery should you take the vaccine?
So the guidance from the UK drug regulators is that there should be a minimum interval of 4 weeks between either having your PCR positivity or the onset of symptoms. The reason for that is that there is still ongoing surveillance of safety related to the vaccine, and you want to have enough time between the infection and the vaccine to see if a fever is likely to be due to the infection itself or due to the vaccine. When we looked at reactogenicity in our trial in people who previously had COVID, who were seropositive for COVID and who were then vaccinated, there was very little difference in reactogenicity between people who are naive to COVID and between people who'd actually experienced COVID themselves. But a 4-week interval is what we suggest from developing disease to being vaccinated.
Thank you. The next question goes to Professor Malik Piris. So, Professor, now you said with the new emergent variants of the vaccine, there may be a chance in future that you need a boost dose. These vector-based vaccines, is there a chance that the efficacy will be low because of the antibodies against the vector?
Yes, I think Maheshi already addressed this issue even with the second dose, and probably she can answer this as well, that there was no evidence of compromisation of the immune response or a boost in the vector antibody levels. And we are talking about a booster dose in 1 year or 2 years' time. I don't think that is going to be a major problem. Certainly not for vectors like the chimpanzee adenovirus, which is the AstraZeneca vector. Now, if you were talking about adenovirus 5 vectored vaccines like the CanSino, because adenovirus 5 is a common virus in humans anyway, then you may be boosting antibody responses. But maybe I'll ask Maheshi if she has any comments on that.
Dr. Maheshi, do you have any comments on that?
Thank you. No, I think Professor Pires is right. As we don't know. Certainly, when we've looked with a short interval between priming and boosting, we've not seen any, firstly, boosting of the anti-Chaddox vector immune responses, and we've not seen any impact on anti-spike responses, anti-spike antibody or T-cell responses. We don't know what will happen if you boost again in a year's time. We have got a subgroup in our trial of participants, a small subgroup of people who've previously had chimpanzee adenoviral vectored vaccines as part of a clinical trial for other diseases. So if we used the same vector for MERS coronavirus, and similar vectors have also been used in clinical trials for HIV, malaria, and Ebola. So we recalled some of those participants who had CHAD at least a year ago, and we immunized them, and we're looking at the data to see whether or not there's any change in immunogenicity with those individuals, and we will hope to publish that shortly.
Thank you. Again, Dr. Ramasamy, is there a difference between what we get in Sri Lanka, the Covishield vaccine produced in India, and the Oxford vaccine available in UK? Do we have a difference? Yes.
So the Covishield vaccine is identical to the vaccine that's been produced in Europe. It's manufactured by the Serum Institute of India under license from AstraZeneca. In fact, one of the things we've done is a batch-to-batch comparison between batches made in the UK and in Italy with the Indian Serum Institute batch. And we've seen no difference in immunogenicity or reactogenicity between those batches. That data hasn't been published yet, but that's given confidence to the Indian regulators to approve that vaccine. So it's exactly the same. You should have confidence in it.
This question goes to Professor Nilika Malavige. Those people who are immunosuppressed or on immunosuppressive drugs, say, suppose a person who has rheumatoid arthritis, only flutamide. What are your advices on receiving of the vaccine?
So actually, because this vaccine, the Oxford AstraZeneca vaccine, that is it is a replicant deficient virus vaccine. So it can be given to immunocompromised people, including individuals with HIV, individuals on any type of immunosuppressive treatment, because those individuals are at more risk of developing severe disease. So that is the reason precisely that the vaccine should be given to them. And also, if you look at the mRNA vaccines, the Pfizer and Moderna, they are mRNA vaccines, and again, they can be given to any immunocompromised patient or any individual on any immunosuppressive treatment. Thank you.
Thank you. Professor Malik Pires, now we are getting the Oxford vaccine to Sri Lanka, the Serum Institute from India, but the media are speaking about the availability of other viruses vaccines in future. Can you compare the Oxford vaccine with the Chinese vaccine and the Sputnik vaccine from Russia?
Yeah, so it's very difficult to compare, because the phase 3 clinical trials are crucial to compare efficacy, right? And the data being published. So as you heard, I think from Nilika at the beginning, that at the moment we have data on 4 vaccines. That's the RNA vaccines, AstraZeneca, Moderna, the Oxford vaccine, AstraZeneca vaccine, and then very recently, as I understand, the Novavax vaccine. Now, the data on the other vaccines, whether it be the Sputnik or the Chinese vaccines, etc., we have heard the data reported, but we haven't seen the actual data. So it's quite difficult to compare. But then also one has to say that even amongst the published vaccine data, it is not so simple as to just take a number and say this is 70% and that is 90%, and 90% is better than 70%, because we are not comparing these vaccines head to head in the same population. We are looking at different populations and sometimes slightly different criteria for endpoint of symptomatic disease, etc. So, I think what we can say is that from the published data, the 4 clinical trials is extremely good, promising, and to be honest, I couldn't have expected more 6 months ago. I would have been glad, delighted to have vaccines of 60, 70, 80, 90% efficacy. So, I mean, I can't say anything more than that, given the lack of published data on the other vaccines. But in terms of what they report, it seems quite promising, but it's left to be seen. Over.
Professor Paris, now the Chinese vaccine being a killed COVID virus, and the emergence of the new variants, how do you see the effectiveness of this virus in the vaccine in future?
So, I mean, the variants, first and foremost, as I told you, these variants are not leading to complete loss of protection. If anything, some of them having a slight drop in protection. So that's the first point, and people really have to, keep that in mind. And so this is not an excuse not to take the vaccine. Now, when you're talking about how the variants can impact on different types of vaccines, these variants that we are talking about, the one found in South Africa and Brazil, the mutations are in the spike, which reduces the antibody binding to the spike and neutralizing the virus. So they will affect all vaccines because all these vaccines primarily rely on neutralizing antibody, and except for the Chinese vaccines, which are inactivated whole virus, meaning all the virus proteins are there, these other vaccines, whether it be the RNA vaccines or the adenovirus vectored vaccines, basically have the spike as the immunogen. So they will impact all of them. I mean, one could arguably, speculate, I mean, I'm only speculating here, that inactivated vaccines, because they do have many other viral proteins which may generate other types of immunity like T-cell immunity, they may have some advantage in that regard. But then, of course, on the other hand, you don't want more proteins injected in a vaccine than you basically need. So in that regard, in terms of adverse effects. So in that regard, the targeted spike protein vaccines, which are the RNA and the AstraZeneca and adenovirus vectored vaccines, are theoretically better in that regard. So I think it's far too early to try to conclude, whether one vaccine may be better than others in terms of tackling these mutant viruses. Over.
Could Dr. Ramasamy elaborate on this? Deaths in Norwegian elders' homes. And there's some news about Indian healthcare workers dying after receiving the vaccine. And in Germany, the people over 65 years, it is recommended not to take the vaccine, but up to 65 only. What are your thoughts on this?
Thank you. Taking those separate points. So first of all, the question about the Norwegian deaths. So this was press reports of several people in Norway who were, as I understand, terminally ill and very frail, who received the vaccine, who sadly became unwell and died soon afterwards. Now, I don't know the details of those cases, but my understanding of that is that they were very, very vulnerable, very frail, highly comorbid individuals. What we see with all the vaccines, the Pfizer vaccine, the Moderna vaccine, and our vaccine, is that people do have reactions afterwards. They have headaches, they have fevers, they have these systemic symptoms. And you could imagine that if you were a very frail elderly person, you developed a very high fever, that might push you into atrial fibrillation and heart failure. You could see how that might happen. in our studies, we've not seen any safety concerns amongst the elderly, and indeed our vaccine was better tolerated by the elderly than younger adults. So that's the first one. The second question about the deaths in India, I have to say I don't know about that.
It's just in the papers. It's just in the
Okay.
papers that there had been 9 deaths in India, and 6 men and 3 women have just died after this. But the Indian the regulators have said it is nothing to do with the COVID vaccine. So, but they have not released any data on the investigations that has happened. So we are not aware of anything about that yet.
Yeah, I have absolutely no knowledge of that, I'm afraid. I mean, what I would say though is that as you start vaccinating millions and millions of people, as we have been across the world, you know, in those populations, unfortunately, bad things happen. You know, people become ill, people have other conditions that happen, and it's really important that we have robust safety reporting so that we can look at each of those cases to see actually, is there any link at all to the vaccine? That's going to be really important for vaccine confidence and to encourage people to take the vaccine. We've got to debunk these myths going forward, and we have to have the safety data out there. The final question was about, I think, Germany.
In Germany.
Not approving the AstraZeneca vaccine in the over 65s. So our efficacy data that we generated in the UK, Brazil and South Africa was based on people recruited in those countries. We had to recruit a minimum of 20% of people who were over the age of 55. That was a requirement by the drug regulators, precisely to look for effectiveness and immunogenicity and safety in the elderly, the more vulnerable groups. And I have to say that we, despite recruiting elderly people, we didn't see a lot of cases of COVID, certainly in the UK, in the elderly. And that's because with all the lockdowns and with the advice from the government, they were all shielding. So they were staying at home, they weren't going out, they weren't being exposed to COVID-19. So they weren't catching the disease. And so if there's no transmission of disease in that age group, you then can't look at efficacy in that age group. So we had very limited data. We had very few cases of COVID in the over 55s. However, what we did show was that the vaccine was equally immunogenic in the elderly. So it had generated neutralizing antibody responses and T-cell responses that were very similar to younger adults. So there's no reason to suppose that the vaccine would be less effective in elderly. We just haven't been able to generate that data in our trials. The German decision is based on that. It's not that there is no efficacy in the elderly. It's likely that it's very similar to our efficacy in younger people. We just haven't been able to generate that data because our elderly have all been shielding.
Thank you very much. And Professor Neelika Malavi is kicking to answer that question on the Indian deaths. She's ready with the answer.
Yes, so I actually, because about these 9 deaths, I emailed the vaccine head, one of the people leading the vaccine trials in another trials rollout in India, Professor Gagandeep Kang. Most of you have heard probably. So of these 9 deaths, there had been 9 deaths and 16 hospitalizations, and they are saying that none is related to the vaccine because except for one which was due to sepsis, the others were acute cardiac events. Which were in the 50-something age group. So this is why they don't think it's because of vaccine, sepsis and acute cardiac events when they have immunized millions of individuals already. This is her answer. Thank you.
Thank you very much, the speakers. Yes, sorry.
This question to Professor Ramasamy. Is in the low dose, standard dose group, was there a better immune response and a protection?
Dr. Ramasamy, did you hear the question? The question is, in the low dose standard group versus the standard dose, was there a difference in the protection?
There was, and that's where that's what the 90% efficacy that we apparently saw in the low dose standard dose group. That was a post hoc analysis of the overall data, and that's a problem with post hoc analyses. You know, if you do lots of analyses, you can get lots of different answers, and it's important to look at the data objectively and carefully. When we picked that apart, actually, it wasn't due to a difference in dose per se. The reason that we saw this apparent difference in efficacy with the low dose standard dose group was actually due to dose interval, because those individuals happened to have a longer dose interval between their prime and boost. And that's what we think is going on. It's actually not the difference in dosing. As I showed you in my previous graphs, when you looked at low dose versus standard dose across different age groups, we actually didn't see much difference at that point. When it comes to the phase 3 data, we think that actually the difference between those groups was due to the dose interval. The longer the duration between your first and the second dose, the more likely you are to have a more mature immune response. It allows time for germinal centers to involute for you to develop better memory B cells. And that's probably the key. It's not the dose, it's the dose interval.
Thank you. We can take one more question. Dr. Anula Vijayasundar, the past president of CCP and SLMA.
Hello, Maheshi. Right. In view of what you mentioned about the higher antibody response occurring at 12 weeks, that is isn't it better than to postpone the second dose of the vaccination to 3 months, to 3 to 4 months, rather than giving it early, where you can get a lesser antibody response? Or when you wait with the chance of getting infection in areas of high prevalence, do you still think it is better to give it early or postpone for 3 months?
Thank you. So the decision that we've taken in the UK is to give as many people as possible that first dose. And we have some limited data that I wasn't able to show you today, but we've done some calculations looking at efficacy post first dose. And it's again of the order of about 70%, and that's looking up to a 12-week interval. So, of course, individual countries have to make their own decisions. In the UK, we are in the throes of a pandemic, and we've got huge numbers of cases in hospital and lots of community transmission. So we've taken the decision to give that first dose to as many people as possible, and then to elect for equity, really. And then for that second dose to come in 12 weeks later, also hoping that will, in the longer term, give you better, more robust immune responses.
Thank you very much, ladies and gentlemen, and thank you very much the experts, Professor Neelika Malavige, Dr. Mahesh Ramasamy, and Professor Malik Paris. We'll have to move on to the next session. The next speaker is Dr. Harsha Satish Chandra, who is a consultant physician at the National Hospital of Sri Lanka, the President-elect of Sri Lanka College of Internal Medicine. Will be speaking on the current therapeutic options, therapeutic interventions in COVID-19. Over to you, Harsha.
Thank you, Gul. I must at the outset thank Professor Seneka Rajapaksha, President CCP, and Professor Tilak Jayalat, President SLSIM, for jointly organizing this timely symposium. Now, after 3 very exciting talks on vaccines, I won't blame you for thinking why we should focus on treatment. But unfortunately, COVID is going to be with us for a while. So in this vaccine era, I think the rollout of the vaccine is going to take time, production, distribution, and all aspects of it. So we are going to have COVID-19 infection for some time to come. And as Professor Malik said in his talk, we may need booster doses, maybe even second, third generation vaccines in future in case there are mutants. So it's going to be a long process. And also we don't know for sure whether the vaccines prevent transmission completely. So transmission is going to take place for a while, and severe disease is going to be there with us. And more importantly, we must make sure that the health systems are not overwhelmed by COVID-19 cases, and also that could adversely impact non-COVID-19 morbidity and mortality as well. So it's important to refine our treatment to a point where we have a manageable number of cases where we are able to prevent disease progression so that we can have more patients outside hospitals, and also in case of hospitalizations, to improve them as quickly as possible and get them out of hospitals so that the health systems can function normally. So that's why I think we need to focus on treatment as well. So essentially, severe COVID-19 is a severe viral pneumonia. But, I mean, we've been dealing with severe viral pneumonia for ages, but COVID-19 has some special characteristics where lots of systems are involved. So if you take the respiratory system, there could be ARDS needing prolonged ventilation, and a particular problem with COVID-19 is that we find that patients have to be ventilated for long. Compared to influenza, there was a study in France which showed that COVID patients had to be intubated, had to be ventilated for 1 week longer than influenza patients. And then there can be severe viremia causing direct effects on the organs, like there could be acute kidney injury, myocarditis, and then this concept of the cytokine storm. Although it doesn't happen in every patient, there could be an overdrive of the immune system, which is a result of extreme inflammation and cause lots of issues. Then the thrombotic complications, like acute MI, acute stroke, pulmonary embolism, deep vein thrombosis. There could also be secondary bacterial and fungal sepsis, and all these things could cause multi-organ failure and death. And the point I want to stress here is that no two COVID-19 patients are alike. So it's a heterogeneous patient population where in individual patients, certain pathophysiological processes may be dominating others. So there couldn't be a sort of a fixed and very sort of, there can't be just one approach to managing these patients, and there should be much flexibility. Now, from this slide stems are possible targets to intervene. Of course, I will not be talking to you much on the strategies of oxygenation and ventilation, but I would be speaking a little on the antivirals targeting specific therapeutic windows. Then, of course, drugs to counter the cytokine storm. Then anticoagulants, because COVID-19 has been highlighted as an illness where thrombotic complications have been many. And then convalescent plasma and even synthetically derived monoclonal antibodies, which have shown some promise, but I'll be trying to argue out whether they have actually been useful. And of course, appropriate antibiotics, antifungals, and antiplatelet drugs when indicated. And of course, the general supportive care, I will not be dealing much with that. Now, the evolution of COVID treatment has seen many antivirals being used. Actually, some of the antivirals did not even show in vitro efficacy, but some did. And if you take a drug like favipiravir, that didn't show any in vitro efficacy, but it was trialed out. And there were some drugs like lopinavir, ritonavir, where they did show some in vitro efficacy, but then in trials they did not show much benefit. So the drug that was most promising was remdesivir. Now, this, of course, had shown very clear in vitro efficacy, and it was used in the Ebola epidemic with some success. The dose regimens were different, but the standard regimen was to give a load-in dose of about 200 milligrams intravenously on day 1, and it was followed up with 100 milligram daily doses over a period of 4 to 9 days, so that it would be a treatment course of 5 days or 10 days. And but it was used mainly in patients who were hospitalized, and in the West particularly, when patients were hospitalized, they were already hypoxemic. So the drug data on early disease is minimal. So it's widely used in the US, less so in the UK and EU. Then it's used in the US based on this trial. Actually, FDA granted approval for use of remdesivir based on this trial. You may have seen this article in the NEJM. I don't want you to read this, but just to highlight that it was based on this study which was published in November. And basically, they had given remdesivir to about 500 patients and placebo to another 500. And they showed that treatment with remdesivir had actually shortened the duration of hospital stay. So in A, the one on the left, top left, is the overall data, which shows that the two lines are separating out, the remdesivir in blue and placebo in red. And what it shows is that the hospital stay was reduced by about 4 to 5 days. And it is the same with in slide B, right top, where patients were not receiving oxygen. And in C, that's a slide where patients received oxygen. So in all those three, I think it was clearly shown that there was a difference. But when it came to patients who were on NIV and high flow oxygen, and right down, in the case of patients who were ventilated, the separation was less. But overall, there was a shortening of 5 days of the treatment the patients had to receive in hospital, but must stress that there was no endpoint for survival. So there was no survival benefit shown in this. And the plot here will show it further. But they showed that in different race groups and and in both sexes, there seemed to be some effect on the shortening of the duration. So they concluded that remdesivir is superior to placebo in shortening the time to recovery in adults who were hospitalized with COVID-19 and had evidence of a lower respiratory tract infection. There was no mention on survival data. And this was the basis for the use of remdesivir in the US. But of course, it's a very costly drug, so not many countries used it. And close on the heel of this study, the WHO Solidarity Group published a study which showed actually a contradictory result. And this was a larger study where they had given remdesivir to about 3,000 patients. And there were certain other arms as well where patients had received hydroxychloroquine in about 1,000 patients, then lopinavir in about 1,500 patients, and some others as well. In this study, I will not go into details. Their conclusion was that remdesivir, hydroxychloroquine, lopinavir, and interferon regimens had little or no effect on hospitalized patients with COVID-19, as indicated by overall mortality, initiation of ventilation, and duration of hospital stay. So in the UK and Europe especially, they did away with the use of remdesivir on a regular basis. Of course, I think some trials are still going on, but it's not standard practice. Then we come to the most important part, steroids in COVID-19. Now, from the beginning, when the illness first got hold in Wuhan, steroids in various forms have been used in severe COVID infection. But there weren't actually properly designed randomized trials that reported clear benefit, although there were anecdotal sort of support for steroids and also even observational studies. So the recovery trial group from Oxford, I think Maheshi might be able to tell us more if she's around, they used a dose of dexamethasone, which is a very simple, widely available drug, 6 milligrams IV or oral for a period of 10 days, and they gave it to patients who needed oxygen and also to patients who did not need oxygen. And I would just, this was the paper, again, they published this in the NEJM, and they had given dexamethasone to about 2,100 patients, and 4,000 patients were given the usual care. Now, in this trial, this was actually, the endpoint was mortality, so it's directly relevant to us. And here in slide A, I think you'll be able to see that the group that received dexamethasone, in the group that received dexamethasone, the mortality was significantly lower. There was a statistical significance. P-value was 0.001. And they had then analyzed the subgroups. In patients with invasive mechanical ventilation, the separation was even more. And also in patients who received only oxygen, still there was a difference between dexamethasone and usual care. But when they did not receive oxygen, the difference was less pronounced, and it did not reach any statistical significance. So their conclusion was that in patients hospitalized with COVID-19, the use of dexamethasone resulted in lower 28-day mortality among those who were receiving either invasive mechanical ventilation or oxygen alone at randomization, but not in those who did not need oxygen. So I think that stands to reason, because patients who do not need oxygen probably are in the early stage of the disease, and giving a steroid to them might even dampen their immune responses and be counterproductive as well. Moving on to convalescent plasma in COVID-19. Plasma from patients who have recovered from COVID-19 has been used, in many illnesses, and it was tried out in SARS as well, and again now in COVID-19. So the aim is to use the neutralizing antibodies in convalescent plasma to counter the virus until the immune system of that particular patient is able to mount a good response. So ideally, the antibody titers in the convalescent plasma should be high, and they should be given early as well. So the US FDA has given an emergency use authorization for that. Then studies are now gradually reporting. There was an RCT, a randomized controlled trial in the NEJM on the use of convalescent plasma in COVID pneumonia. There they had enrolled patients who were hospitalized for hypoxemia, and the only thing was that they gave the convalescent plasma at a median of 10 days, so it was not very early on in the illness. And the antibody titers were quite high, 1 in 3,200, and the primary outcome was the clinical status at 30 days. And actually the mortality was not different in the convalescent plasma arm and in the placebo arm. So there wasn't, the conclusion was that there was no significant difference, but of course the intervention was rather late. And there was close to home, there was a trial in India as well. They named it PLACID, and they compared the convalescent plasma and standard care with patients given standard care alone. And the dose that they had used was about 200 ml of convalescent plasma given 24 hours apart. The main outcome they assessed was all-cause mortality at 28 days. And there also they had not seen a difference. And there were some other trials as well. There was one small trial from Argentina which had shown some benefit in elderly patients. But one thing going for convalescent plasma is that the side effect profile is not so bad. It's less than 1%. So That's probably why trials have been going on. And then this new thing, monoclonal antibodies in COVID-19. Now, this is given as an infusion. There are several drugs which have been given emergency use authorization by the FDA. One such drug is bamlanivimab, which is directed against the spike protein. And then there's but they had wanted to give it to prevent progression of COVID-19 in patients who are really vulnerable. So, and it was actually given in an out-of-hospital setting. And a second antibody cocktail from which was called REGN-CoV2, which was a combination of casirivimab and imdevimab. And that was shown to reduce the viral load in non-hospitalized patients. And as a matter of interest, this was the antibody cocktail that the former President Donald Trump had received in his course of treatment. So this has some promise in the treatment and also in post-exposure. Now, anti-inflammatory therapies are also an important part. If you go back to a slide where inflammation was shown to be of importance, and this is to treat the cytokine storm. There was a trial published in the NEJM quite recently, 7th January, and there they had used tocilizumab in patients hospitalized with COVID-19 pneumonia. And I would come back to that in a minute. And then there was another unpublished REMAP CAP study done in which was reported on by UK, and that seemed to show some beneficial effect in certain subgroups. There are lots of other anti-inflammatory drugs that are used. Going back to that trial I mentioned earlier, this is the trial of tocilizumab, which appeared in the NEJM, but their conclusion was that in patients who were not receiving mechanical ventilation, tocilizumab reduced the likelihood of progression to the endpoint of ventilation, reduce the likelihood of progression to the composite outcome of mechanical ventilation or death. Now, anticoagulation in COVID-19 is very important because these patients have extreme inflammation, and extreme inflammation is a prothrombotic state. And actually, postmortem studies of COVID-19 are few. One study from Italy showed that in a group of 20 cases, microthrombi in the lungs had been there in about 60% of cases. But some patients actually have pulmonary embolism and DVT as well. So when patients become hypoxic, it's rational to start them on low molecular weight enoxaparin, and as for prophylaxis of VTE, the usual dose would be 40 milligrams subcutaneous, and in patients with renal failure, heparin is substituted. So if D-dimer is over 1,500, or TEG shows a hypercoagulable state, or if you have strong clinical suspicion, then enoxaparin is converted to a treatment dose of 1.5 milligram per kg subcut daily. Now, there was this interesting concept that aspirin might play a role to reduce thrombotic complications of COVID-19. Actually, in a retrospective study done in Maryland and published in the journal Anesthesia and Analgesia, they showed that they had studied 98 patients who were on aspirin and 314 patients who were not on aspirin. And the patients in the arm that aspirin was given, the patients were 43% less likely to have ICU care, 44% less likely to have invasive mechanical ventilation, and 47% less likely to die in hospital. But of course, this was an observational study, and no RCT has reported so far. And there have been studies as well. In Sri Lanka, we give aspirin, or we continue aspirin in patients who have high cardiovascular risk, or we commence on aspirin when they have, once they are diagnosed with COVID. There has been some talk about ivermectin, which is a drug, antiparasitic drug. Actually, ivermectin had demonstrated very high in vitro efficacy. Actually, it's said to be more than that of HCQ, but the trial data is not very robust. Only small RCTs have reported some benefit. There have been some observational studies. There's one particular study which was done in Florida, USA, and reported in the journal Chest. So there they had actually shown that patients who were given ivermectin actually had the mortality benefit, but then in that particular arm, steroids were used in high dose. So it was a confounding variable which they couldn't sort of interpret properly. And I think RCTs would be needed to decide on the issue. But there has been a change in the US FDA approval. Actually, they had initially said that ivermectin should not be used in patients with COVID-19, but now they have changed their recommendation to say that they have removed the recommendation against ivermectin and said that it could be used in trials. So what happens to a patient when he gets COVID-19 in Sri Lanka? So asymptomatic patients and even mildly to moderately symptomatic COVID-19 patients who are diagnosed using PCR and the RAT are isolated in either in intermediate care centers or in hospitals. And those patients are given low-dose aspirin if their cardiovascular risk is high, of course, only the adult patients. And then patients with multiple comorbidities, and if the severity is more, then they are transferred out to designated specialized COVID hospitals. And in those hospitals, whenever a patient's saturation drops, they're monitored carefully. When the patient's saturation drops below 94%, we do start them on IV dexamethasone as per the recovery trial data. And of course, VTE prophylaxis is also started with enoxaparin in that instance. And of course, when the oxygen need increases, the patient is transferred out to HDU or ICU facilities, and they are given various modalities of oxygen treatment, and I will not discuss that. And again, as I mentioned earlier, the VTE prophylaxis dose will be converted to a treatment dose at a very low threshold. And dexamethasone is continued for about 10 days. I think Ananda will probably elaborate more on the management of actual cases in Sri Lanka. And so I think I would wind up there and hand over to Dr. Vijay Vikrama to present the next lecture.
Thank you, Harsha. The next speaker is the former president of the Ceylon College of Physicians, Dr. Ananda Vijay Vikrama, who needs no introduction. He works as a consultant physician at the National Institute of Infectious Diseases since 2003 and undergone postgraduate including dengue, SARS, avian flu, and other emerging infections and HIV. Research interests include clinical management of dengue, HIV, and chickenpox. He is a member of the editorial board of the guidelines for the management of dengue fever and DHF in adults in Sri Lanka. He won the PROMED Award for Emerging Infectious Diseases in ICID in 2016. Over to you, Ananda.
Yeah. Okay. I think today the main interest is regarding the vaccine. So I think my talk will be brief. I thought of discussing about few management issues related to COVID patients. Before that, I would like to make a statement. That is to say that I had the Covishield vaccine today. I thought of mentioning that because in the morning I had many calls from my colleagues asking whether it is safe to take it. The safety had been assessed by several clinical trials, and the National Medicines Regulatory Authority of Sri Lanka has the expert committee of that has gone through the safety data as well as efficacy data and has recommended its use because the experts who assessed that thought it is very safe as well as reasonably effective. That is for the what we had today. We had our first patient admitted to National Infectious Diseases of Institute on 26th of January. Since then, we had more than 3,000 patients, and we had around 200 ICU admissions, and we had 55 deaths. However, the biggest challenge, I think, was to ensure the safety of healthcare workers, because the confidence in healthcare workers and assured safety is very important to continue proper patient care. And that, of course, has been highlighted in this article published in The Lancet. It says no patient safety without healthcare workers' safety. And as such, we take, in addition to having basic safety measures like hand washing, practicing cough etiquettes, maintaining physical distance, and so on. And also, we try to use PPEs as appropriately and correctly. So, yes, one of our nurses wearing the full PPE kit, then attending to a patient, taking blood. And while wearing these, we do necessary things that give necessary care to the patient. This is when we were aspirating a pleural effusion of a COVID positive patient. That was, of course, he had coexisting tuberculosis, which was later found to be. Just to illustrate a case history, this is a 48-year-old Mr. MS admitted just a couple of 2 weeks ago with the history of fever and cough for 5 days. In addition, he had typical features of COVID like anosmia, loose stools, and hemoptysis, and he had a past history of diabetes too. And on examination, in fact, he from home, he came in 1990 ambulance to the hospital directly. And on admission, he was dyspneic, he was tachypneic, and with low saturation, in spite of giving 15 liters initially. At the beginning, his saturation was 93%. And blood gas, he was tachycardic. ABG showed a pH of 7.36 and PO2 of 32. Then we move into the ICU, and these are his investigation results, which are quite typical of problematic or severe cases of COVID. You can see the hematocrit was stable, hemoglobin was stable, and the white count has gone up slightly. And you can see the typical neutrophil leukocytosis, 82% neutrophils, and then going up to 89%. And the lymphopenia, which went down to 5.4. And many patients with complications tend to get thrombocytosis. This patient, it went up to 730, as Harsha mentioned in his previous talk. We start these patients on aspirin, as this is one of the complications, and his CRP was initially quite high. Again, a feature of COVID, but this tend to confuse us with the secondary bacterial infection, which we don't know, and some patients may be having a secondary bacterial infection too. So such patients will start on empirical antibiotics because we are not sure whether the rise of CRP is purely due to COVID or due to coexisting bacterial infection. Liver enzymes are slightly high, IL-6 was little elevated here, and this was his chest X-ray on admission. You can see bilateral shadows, typical shadows of COVID pneumonia. He was treated with dexamethasone 6 mg daily. IV ceftriaxone started on empirically after blood culture and with oral clarithromycin. we started him on enoxaparin. And later, because the cough was continuing and respiratory symptoms continuing, after about a week, we added him on oral fluconazole 2. And he responded to non-rebreathing oxygen via non-rebreathing face mask. And then we continued it with high amount of oxygen for the first 3 days. And then we tried to reduce it to 70%, but then again he started desaturating. We had to increase it again back to 80%. Then slowly we were able to wean him off oxygen. Today we are moving him out of the ICU. He is now without oxygen. He can manage. Still he is a little bit dyspneic on exertion, but now he is quite stable. So such patients we see, but the reason I thought of discussing about this is this patient came from home directly to the hospital. So with the increasing number of cases, we might see more and more patients like this. And I think as clinicians, we have to be aware of this, and also as doctors, we should aware that other people might present like this directly to the hospital. And if they are at home for too long, I think that can give rise to problems. I think that is probably something which is happening in some countries, and that may be the reason for increased death rate we see in some countries where people cannot be admitted. Everybody cannot be admitted. I don't say everybody should be admitted, but the symptomatic patient should be under monitoring system. This was his chest X-ray 3 days ago. You can see the improvement in the chest X-ray. This is a chest X-ray of another patient, a 48-year-old male, admitted last year. You can see the X-ray showed few changes. He was with dyspneic on admission. He did not have any other comorbidities. And then, because he was dyspneic, we took him to the ICU, started him on oxygen therapy. That time we were not giving him enoxaparin because there was no thoughts on this thrombosis at that time. This was March the last year, and you can see how the X-ray was getting worse. This was X-ray on 27th, and he's being intubated and being ventilated, and then did the X-ray on 5th of April. Yes, ups and downs, but then started to deteriorate after ventilating him for about I think about 2 weeks. This is the X-ray on 7th. The right lung is almost gone, and a lot of shadows on the left lung too. And then he succumbed to the illness. We could not save him. So the challenge is with what to treat. There's guidance given by the WHO Therapeutics and COVID-19 Living Guideline. It changes frequently, and as I was privileged to be in the panel of this together with Shalini Ranganathan from Sri Lanka, and the latest recommendations says that the strong recommendation against the use of hydroxychloroquine and lopinavir and ritonavir, regardless of disease severity. And it further goes on to say there's strong recommendation on systemic corticosteroids and conditional recommendation against systemic corticosteroids in patients who are not hypoxic. And then again, there are conditional recommendation against remdesivir in hospitalized patients with COVID-19, because as Harsha mentioned, even though the trial showed some benefit in shortening the hospital stay, there was no definite survival benefit or significant benefit. So the recommendation after analyzing all the evidence available, that was the decision of the panel. So it is interesting to note regarding the recommendation of dexamethasone, which was based mainly on the recovery trial. And as Harsh mentioned, the evidence is for the benefit for the patients who need oxygen, and those who do not need oxygen, it is not beneficial and maybe may cause harm too. So therefore, it should be started only on patients who needs oxygen. However, having said that, when you look at the data, you see even in the treatment group of this trial, in patients who had dexamethasone, still they had a mortality, quite high mortality rate of 22.9. The control group had, of course, a 25.7 mortality rate. So there was a significant difference with a p-value of more than 0.001, less than 0.001. Still, this amount of this degree of mortality rate, probably the dexamethasone was effective in certain subgroups. So therefore, I think we have to give targeted treatment. I think we will have to wait for the subgroup analysis of this study. And at present, we start hypoxic patients on dexamethasone. And also we must say that at the same time, we have seen issues due to that also. A lot of people who are not diabetic get hypoglycemia, and diabetics, it goes sometimes to uncontrollable levels. And also it may be contributing in some patients, especially those who are on ventilator, for secondary bacterial infections and secondary fungal infections. And the recommendation on vitamin D, I thought of showing that because there's a lot of interest on vitamin D. The NIH and other guidelines, NICE guidelines and so on, do not recommend vitamin D as treatment of COVID-19. This patient, as Harsha mentioned, there's a huge diversity in these patients when we see these COVID patients. This patient was transferred from base hospital Samanture. He was admitted to Samanture Hospital on 9th of January and transferred to us next day. He was admitted there. He had a history of fall on 28th of December. He was AFL sick since 1st of January and was confined to the bed. And he has a past history of ischemic heart disease, diabetes, and hypertension. And he was on admission to Samanture Hospital. He was tested for COVID and found to be positive. And then since he was disnicked, he was transferred to us. When he came to us, his GCS was 7 and the respiratory rate was 40 per minute. We did a CT scan brain that showed the normal pressure hydrocephalus. There was no infarctions. And because of his low GCS, we had to intubate and ventilate him. And this was his chest X-ray, which was basically normal. And the repeat chest X-rays too were normal. However, his conscious level did not improve. We had to continue him on ventilation. And around 15th day, we decided to do a percutaneous tracheostomy. This is, you can see we were doing the tracheostomy in our ICU. It's difficult to get others to do it, so we have started doing that now. And so it was, even in spite of all this, he succumbed. His conscious level did not improve, and then he succumbed to his illness. So he did not have COVID pneumonia as such. We could not get an MRI. We were not sure whether he had some insult causing to the brainstem with that fall, or whether he had some brainstem infarct and had the fall and remained immobilized with low GCS since then. This case history just to show the diversity of the presentations with COVID. This is a 94-year-old male transferred to us on 3rd of January from a private hospital with a history of cough and fever and with body aches. And he was in spite of his age, he was an active person, but with this illness, he was finding it difficult to walk, which was gradually progressive. When he came to us, his saturation on air was 92%, but then he improved with low dose oxygen. He was hemodynamically stable. Later, he became confused. Oxygen saturation maintained with 2 liters of oxygen. His blood counts, he was C-reactive protein was quite high, and you can see he did not show typical lymphopenia, and platelet counts are quite stable. Liver enzymes a little high. His creatinine was high, and the chest X-ray showed patchy consolidation in all 3 zones of right lung and mid-zonal and perihilar region in left lung. and it was reported as could be due to COVID-related lung changes. This was his chest X-ray, and you can see the progression of the illness. It was improving, and his CT scan brain, which was done because he was confused, showed only age-related cerebral atrophy and small vessel disease and mastocytitis with sinusitis. And the chest HRCT showed ground glass opacities with associated patchy consolidation in both lungs, suggestive of COVID lung changes. In addition, he was found to be having nodular opacities in both lungs with bone destruction, suspicious of a neoplastic change. In spite of all this, he was given oxygen via nasal prong, self-proning, IV ceftriaxone, or Clarito, enoxaparin low dose because of his renal impairment, and he was given ectopic insulin for his sugar. And in spite of all the problems and the age he had, he recovered and went home 2 weeks later. His antibodies became positive, he gradually improved. And so in spite of all the problems, he went home. So it is real diverse picture when it comes to these patients. And so the challenge is with what to treat. And as Hansha mentioned earlier, we were thinking of more of pneumonia illness, but now the pathophysiology, at least partly played by thromboembolic events or microvascular thrombosis of the lung. And the prevention of that is important, and still the recommendations regarding low molecular weight heparin varies from the guidelines vary from country to country. We have started using this on hypoxic patients, and in critically ill patients, I think it is considered the ECMO and embolectomy is consideration of that is recommended. So still it is a challenge with modes of treatment and also with the still limited understanding of the pathophysiology. This challenge tend to remain, and in spite of large number of clinical trials, some are concluded, many are still continuing. In spite of all this, still we are yet to find a definitive treatment. Ventilator strategies have changed. Tone ventilation has shown a significant improvement in oxygenation of these patients, even in awake patients who are not ventilated. Self-prone position is advised, and the oxygen treatment is probably the only proven method of, or may not have evidence, but the only mode of treatment available. And of course, we have to emphasize on non-invasive ventilation. When it fails, of course, we'll have to do invasive ventilation. But many patients seem to be responding to non-invasive ventilation, like high flow nasal oxygen and even oxygenation with non-rebreathing face mask. I think therefore we have to advocate having more facilities on such interventions like non-invasive ventilation and oxygen treatment, rather than stressing too much on ICUs, because I think many patients respond to this treatment. So in addition to treating this, I think probably our responsibility comes to getting involved in trials too, because as we all know, as I said earlier, still no treatment has been found for this condition. Thank you.
Thank you, Ananda. We have come to the end of the sessions today. Let me invite Professor Sendra Rajapaksa, the President of Ceylon College of Physicians, to wind up the sessions.
Thank you very much, Wool. That was a very interesting session which covered practically everything there was to cover about COVID-19. So very special thanks to the three speakers on it was a kind of symposium on the vaccine. Dr. Ramasamy, Professor Malik Piris, Professor Malivage. Special thanks to three speakers. We know a lot more about the vaccine now. And also Harsha, Dr. Satish Chandra, my good friend, and Ananda, thank you very much for your excellent talks. There are certificates of appreciation which, is there a camera here? Yes, they can see it. For all the speakers, which I will virtually hand over, but you'll get the electronic copies. And a few other people to thank for this, for making this a success. First, of course, Dr. Puldisanaik for his typically brilliant comparing and moderating. Dr. Madhuvanti Hettiarachchi from the Sri Lanka College of Internal Medicine is the one who organized it and did all the hard work in finding the speakers. We'd like to thank all the participants also, and from the ground team, Zydus Cadilla for sponsorship, and ClinMark staff and the audiovisual team for all their support. So thank you very much for attending. Thank you.